Dear learners of NIOS, today we are studying biology, shoot system 2. So, before I start today's discussion, I would like to recapitulate the previous part of it, that is shoot system 1. In that we had studied that the aerial part of the plant is the shoot system, and it is erect, generally goes away from the geotropic pull, and it is towards the sun. The shoot system grows towards the sun. So, these were the qualities and the kind of organs that a shoot system bears are leaves, buds, fruits, and lateral branches, and how we had also discussed how shoot system is different from the roots, not only for the external characters, but also for its anatomy, that is internal structure, right. So, we shall be moving on to the internal structure of the stem. Now, since we understand that the stem and the roots, they too are quite different from each other, and their structure and functions correlate. So, whatever structure the stem bears has its own purpose and function. So, there is always this kind of relationship in most of the living organisms. So, plants are also living, and they have this structure and function relationship. So, the internal structure of the stem can be studied if we cut the stem transversely and observe it under a compound microscope. So, this is the compound microscope, and I am going to cut one section of the stem, and in the laboratory you can always use very fine or new blades to cut the stem and prepare a slide with the help of your teachers. So, for example, I have this stem with me, and we try to cut the section. See, this woody character is because of the secondary growth in the plants. Secondary growth we will discuss later, and now you see the precision, the fineness of the knife, which helps me to cut a very thin slice of the stem, and we try to observe it under the microscope, and the structure generally temporarily it can be observed like this, but generally what we do is we color it with various reagents, and all the tissues which are either hard or soft, they can take up their separate colors. That is how the all the cells, they take up different colors of stains, and under the microscope we try to observe. So, in this, when we observe what we are seeing, we have to focus it as per the number of my own eyes focal length, and that is how you also adjust it and set it to the structure we are trying to see. And under the microscope, what we get to see, I will try to talk about it. First, I will show you one picture. This is the picture, and what structures I see under the microscope, they shall be visible on your screen now, and I will try to tell you. This is a diagrammatic diagram and not exact picture of the microscope, of the slide, but with this help we will try to understand the structure of stem of a dicot plant. So, what we see in the diagram of the slide, first layer, which is labeled as number 1, is epidermis. It is the outermost layer. Then number 2 is the hypodermis. Hypodermis is the few cells which are thin layered, and they are below the epidermis. So, epidermis is the outermost covering, and scales you are seeing, they are multicellular hair-like projections which are called scales. You remember, in case of roots we have unicellular hairs, but in case of stem we have multicellular. You can see the division of cells, and so many cells make one scale. Then number 3 is the general cortex or the soft tissue or the parenchyma. Then number 4 is endodermis. Endodermis marks the special layer which demarcates the outer portion from the inner vascular structures. Endodermis generally has thick wall, and there are few cells which have thin wall. They allow the passage of fluid from them. So, number 5 is sclerenchyma, the hard bast, number 5. Look at it specially because the cells appear darker here in the diagram, and actually the reason is the wall is very thick. So, they provide a stronger structure and cover for the delicate tissue beneath, and the delicate tissue beneath, number 6 is phloem, and number 7 is the cambium, which has the meristematic activity of forming new cells in the secondary growth of the plant. And number 8 is number 8 and 9 both are actually xylem, and number 8 is metaxylem, the older xylem vessels and tracheids actually, and number 9 is the protoxylem, that means the fresher xylem vessels, all right, tracheids. So, number 10 is the wood parenchyma, which is at the center, and that makes the pith of the stem. So, the girth of the whole stem is given by the pith. It is the soft tissue which is central in position in the stem. So, I would like to show you one specimen here. If you focus on this and if you try to see the natural presence of, this is the cortex on the periphery, the dark green which you get to see on the periphery is the epidermis, and this is the central pith, all right, and here is the xylem and phloem. So, when I cut the section, I cut it transversely. If I cut a long section like this, then I call it vertical section, all right. Can you see the vertical section, how I cut it? It is like this, okay. So, if I cut it like this, then it becomes the transverse section. So, right now the diagram that you are seeing is the transverse section of the stem, okay. So, this is how we understand the internal structure of the stem. Most of the dicot plants, they have this kind of structure. Now, we talk about medullary rays, which is the narrow margin of parenchymatous cells between the vascular bundles. If I go back to the same slide, do you see that there are two bundles, and in between there is a layer of normal cells, and that normal parenchymatous cells, they are separating the two bundles. So, this particular parenchymatous layer of cells between the vascular bundles is called medullary ray, all right. So, medullary rays are the narrow regions of parenchymatous cells in between the vascular bundles. Next, last is the pith. I showed you pith in the sample. So, the central parenchymatous zone with the intercellular spaces, they are loosely packed cells, they are not hard packed cells, right. So, now let us move to the monocot stems. You understand the difference between dicot stem and a monocot stem. So, earlier we discussed about the dicot stems, now we are going to talk about monocot stems. Monocots are represented by family Gramineae, generally grass family, and the representative samples that you understand, the examples are wheat, maize, garden grass, etc. So, their stem structure is quite different from the dicot stems. Generally, we see that monocots are short-lived plants. They grow for a season, give off their seeds or their product, and they die off. But dicots are the plants who live longer, and they have to undergo secondary growth in the stem, so that stem grows in girth. Over a period of time, you must have observed that a small plant which you ever planted in the garden has become thicker, thicker, thicker and thicker. So, this is how secondary growth is necessary for the dicot plants, because they have to live longer and for many years together, and they have to be stronger to understand the to withstand the atmospheric or geographical changes. So, this is how main difference in the life of monocots and dicots is, hence it influences the stem structure as well. A transverse section of monocot stem reveals the following structures. So, the outermost is again same epidermis, and in the center is the ground tissue. It is a mass of parenchymatous tissue, and only a few peripheral layers below the epidermis are sclerenchymatous. Now, we must understand the difference between parenchymatous tissue and the sclerenchymatous tissue. Parenchyma is thin-walled and loosely packed tissue, so it does not provide strength, but sclerenchyma has thick-walled tissues and it provides strength to the stem, and this is called hypodermis. All right. So, vascular bundles in monocots are numerous, scattered in the ground. In case of dicots, if you remember, they were arranged in a circle, right? But in case of monocots, they are scattered under the in the ground tissue. So, each bundle has its own qualities. They are collateral and closed. There is no cambium strip between xylem and phloem. You remember, cambium is the special tissue which has ability to divide the cells. So, now since monocots do not need secondary growth, so they do not have much of cambium between xylem and phloem. So, all right. So, endark xylem and xylem occurs in the form of letter Y, and the inner most protoxylem is disintegrate to form a water cavity, so that the conduction of water in the stem becomes a easier thing for the plant. So, we'll see this diagram now, and you can make out in the very first diagram, which is labeled as A. It has so many vascular bundles spread in the ground tissue, and if you look at the figure B, you can further see a bigger version of it, magnified stem TS of the monocot, and in this you are able to see a little bit of nature of the cell wall and the kind of tissue involved. And in diagram C, if you see, there are metaxylem, there is protoxylem and Y-shaped arrangement of the xylem. Then there are companion cells and sieve tubes which represent phloem. Sieve tubes are a component of phloem tissue, and the bundle sheath which is called sclerenchymatous in nature. Now, you understand sclerenchymatous, I already told you. So, secondary growth, why now let us move on to why secondary growth in the stem is required. I already told you that plants with age, they need to add strength to the stem, so that the stem can bear, or the tree or the plant can bear the pressures of air and other atmospheric like rains and typhoons and hence they have to be stronger and keep the plant upright for many years, hence the secondary growth is required. This is why you can see that very tall trees can withstand strong winds and lashing rains without falling down. But monocot plants like wheat, rice, maize and grasses, they get easily washed off because of there is no secondary growth in their stems. So, you get to hear that if there is heavy rain in the wheat crop, then there is lot of loss happening and farmers are really suffering that loss, because monocots have weak stems and they fall down because of the rains, hence this loss. Let us move on. The activity of the vascular cambium from the secondary vascular tissue is as follows. The strip of the cambium is in the vascular bundle, it is called fascicular cambium. They are the cells having power to divide. Then now see this diagram, you can easily understand from this. Figure A, you can see a dicot stem and vascular bundles arranged in the form of a ring in figure A. In figure B, second stage has come where the cambium cells, they join from the medullary rays, in between they grow out and they meet the next this layer. And in the third figure C, if you see, there are a lot of medullary rays and secondary xylem which is cut out with the help of fascicular cambium. A new xylem is cut out and new phloem is cut out, and this is how stem gets good range of strong tissue. And outside near the cork or the bark, as you see in this case, I would like to show you this plant. The outer surface which you are seeing, this is the phloem, and cork, it is generally, it has outer layer also has meristematic activity and it adds on cork tissue. Now, when you visit a kikar plant or bhojpatra, then you see that the bark outside tissue can peel off like layers of paper, and that is used as in olden times it was used as paper to write down on. So, this is how internal growth and the external growth, secondary growth adds strength to the stem. Let us move on to lenticels are the breathing holes in the outer cork layer, and phallogen, phalloderm and phloem together constitute the periderm or the cork tissue which we discussed outside. Due to internal increase in the thickness, the periderm replaces epidermis and becomes protective in function, and it is very strong, it is really protecting the stem. All the dead cells lying outside the active phallogen, they constitute the bark. So, do you know dalchini, which is a very famous condiment? It is a bark, and so many other spices are actually obtained from the barks of certain trees. Barks of certain trees are used as medicine as well. So, well, let us move to the TS of an old stem, and a portion has been enlarged, and just have a look and then we will move forward. So, I want you to focus on bark. Can you see number 1 point which is called lenticel? Lenticel is the breathing area of the bark. Here the cells are thin walled and gaseous exchange can happen, and below that is the epidermis and then the cork cambium. So, all these make up the cork. All right. So, inner structures we have already explained how metaxylem and secondary xylem is cut and how the tree stem or the girth grows over the years. So, let us move on to the leaf. Leaf is the flattened and expanded lateral appendage of the stem or the branch which develops from its nodes. We had already discussed what are nodes and what are internodes. So, leaf generally grows on a node. It originates from leaf primordium formed by the shoot meristem. See, if you remember, shoot has its cells on the apex which has meristematic ability. Meristematic ability means they are capable of division and they bear a bud in its axil called axillary bud. From here the leaf develops, all right. So, if you remember, I have shown you this kind of sample where you had seen these are the buds, special buds, and in this such big leaves in due course of time will be formed, all right. So, they can be present in the axil of any leaf or they can be present as a lateral appendage. So, what are leaves? Actually, leaves are the important physiological processes of photosynthesis is carried on in the leaves, and the transpiration and respiration also is done in the leaves. So, besides protective axillary buds, leaves can get modified into certain structures for the storage of food, water, climbing up for the weak stems if you remember, and vegetative propagation. For example, I had discussed adrak or the ginger if you remember. So, if you break one bud of ginger, it can grow into another. So, or even your mint or which you popularly known as pudina. So, pudina also grows like that. So, this is how vegetative propagation means making of new plants from the old plants is also helped with the help of stem. So, right now we are discussing about leaf, the structure of leaf. Now, this is a leaf, right? Can you see the structure? This is the petiole. With this, it is attached to the stem, all right. So, this is the leaf lamina and this is the stalk, all right. So, the leaf base, I will show you in another plant in which leaf base is very specific, all right. Can you see this? This is the leaf base. With the help of this leaf base, this is a rose leaf and this is the leaf base. This is how it is attached to the plant, okay. So, now we will move on to the petiole. This is the petiole. It is the stalk of the leaf. Leaf can be petiolate or without even this stalk. So, such leaves which do not have petiole or the stalk, they are called sessile. Have you seen some leaves directly coming out of stem like this? So, they are called sessile, all right. So, in monocots also sessile leaves can be there. The petiole may get modified and they swell up, for example, in certain plants for floating, they become like orange and filled up. So, these are the, now let's move on to the second feature of the leaves, whereby you are seeing not just green, but there can be many other colors of the leaves, but they all help in performing photosynthesis, which is the main function of the leaf. Now, let us focus on the leaf tips. If you see leaf tips, so leaf tips can be different, and let us see some of the leaf tips. This leaf has one kind of tip, this has another kind of tip, this leaf has another shape, some different one, and this is serrated like this. So, now you see that leaf can have different morphological appearances along with the various modifications for functions, and leaf blade or the lamina is the green and thin flattened part of the leaf, and on that veins and veinlets transverse through its surface. Can you see this leaf? This is the main vein. Can you see the main vein and the side branches? So, this is called veins and veinlets. They transfer through the whole surface, and their main function is to keep the leaf in a spread form. Okay. So, this is how function of the leaf is performed, and most prominent vein which run through the base till the apex is called the midrib. Okay. As I had shown you in this. So, this becomes the midrib. Okay. Right. So, let us move on to one summary, and now I hope the internal structure of the leaf we shall discuss in some other lesson, and right now let me quickly summarize to you that all the dicot and monocot stems undergo certain kind of functions, and monocots generally do not have secondary growth, whereas dicots, which have to live for the years and have to grow stronger, they have secondary growth. Secondary growth in the stem has a particular system to follow, certain steps to follow, and this is how wood is formed inside, and the stems are very strong. They withstand rains and thunderous geographical calamities or whatever. So, this is how the leaves which carry the main function is photosynthesis, and the main vein and the veinlets make the part of the leaf lamina. Leaf lamina performs the major function of photosynthesis. So, now we have covered two important functions carried out in the aerial part of the plant. So, thank you so much for your attention, learners. With this we conclude this lesson.